Good day, this is the Professor, CBMD PhD, and today is Monday, August 22nd, 2022. It is 1:35 and 7 seconds, AM Eastern Standard Time, and today's research, cell nucleus. So let's hop to it here. Unlocking cell nucleus behaviors, deformation mechanics, may be key, and cancer fight. Date August 18, 2022. Source, Texas A&M University. A team has uncovered surprising mechanical behaviors of the nucleus. For years, the nucleus within a cell was thought to be elastic, like a rubber ball, deforming and snapping back into shape as the cell navigated through pores and between fibers inside the human body. Researchers have now discovered that the nucleus is more complex than originally believed, behaving more like a liquid drop than a rubber ball. The discovery that the nucleus deforms like a liquid drop calls for a fresh look at how the nuclear shape becomes abnormal in diseases like cancer. The genetic material governing a cell's function and behavior, called the genome, is safely stored in the nucleus. And nearly 150 years of looking through microscopes has taught pathologists and researchers that misshapen nuclei are warning signs of diseases like cancer. The cancer cells with such abnormal nuclei are able to migrate to other parts of the body in a process called cancer metastasis, a spreading that can be lethal. The nuclear shape observations are used in cancer diagnosis, even today, but why nuclei become abnormal has remained unclear. By understanding how nuclei become misshapen may help uncover a way to aid cell nuclei in regaining their normal shapes, leading to new approaches for treating cancer. The findings from this study are critical to understanding how a protective layer surrounding the nucleus, called the lamina, helps preserve nuclear shape, while cells go through the tortuous path through pores and around tissue fibers. Researchers began their exploration of nuclear behaviors by placing fibroblasts, the most common type of connective tissue cells in animals, into a miniature obstacle, course of tiny, flexible pillars, 1/100th of a width of a human hair. In order for the cells to crawl through the obstacle course, their nuclei had to squeeze in between the pillars. The researchers observed the movements with advanced high-resolution microscope that could image the 3D shapes of the nuclei. The imaging revealed that the pillars created deep indentations into the nuclear surface, yet the overall nuclear shape was preserved, allowing the nucleus to successfully pass like a liquid drop, and unlike a springy elastic rubber ball, through the obstacles. The research also revealed that a depletion of lamin AC, one of the normal protein components of lamina, caused the nuclei to get entangled in the obstacles. The discovery suggests that lamin AC helps maintain the surface tension of the nuclear drop. The work points to a fundamental mechanism by which the nucleus preserves its shape and protects its genome. The discovery also helps us better understand how misshapen nuclei arise in cancer and how to potentially make them normal again. The researchers are now studying the implications of the drop model for the abnormal nuclear shapes commonly observed in cancer. And this is the Professor of Microbiology, Bioinformatics, and Pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.